When you think about Mexican identity,
Mm.
it's just such a rich mix, isn't it? A tapestry woven over thousands of years.
Absolutely.
But how do you really see those layers, the indigenous roots, the Spanish arrival, the African presence,
Yeah.
how they actually combined?
Well, for a long time, yeah, it was mostly history books, maybe family stories.
Right.
But now we can literally read that history in people's DNA. It's quite amazing.
So we're talking about genomics finally catching up.
Exactly. For years, the global map of human genetics had this huge gap, especially over Latin America. But these big projects like the Mexican Biobank and some really impressive work from INEM and Gen, that's the National Institute of Genomic Medicine in Mexico, they've really started to fill that in over the last decade or so.
And they're not just looking at a few people.
Oh, no, this is large-scale stuff. Thousands of individuals. They've looked at both indigenous groups with deep roots and the wider mestizo population, the mixed population, across, I think, almost all 32 states.
Wow. So this isn't just about finding out your ancestry percentage. It's more like reconstructing how populations moved and mixed over time.
Precisely. Pre-Columbian, colonial, post-colonial.
Yeah.
You can see the echoes of those eras in the genome. It's like demographic forensics.
Okay, so let's unpack this then. Our mission today is to trace those dynamics, see how the story unfolds from the very first people right up to the incredible diversity we see today. Where do we start? The deep past.
Definitely. To get the modern picture, you have to go way back. We're talking 19, maybe 23,000 years ago, peak ice age, what scientists call the last glacial maximum, or LGM.
Right.
The general agreement is that the first people in the Americas came over from Asia, crossing that Beringia land bridge when sea levels were lower.
An incredible journey to even think about. And these first arrivals, they laid the genetic foundation for everyone who came after.
That's the idea. And we see it really clearly if we look at the maternal line. That's the mitochondrial DNA, the mtDNA, passed only from mother to child. Research shows there are 4 main sort of pan-Native American haplogroup-specific genetic lineages called A2, B2, C1, and D1. They form the base for almost all indigenous populations across North and South America.
Okay, A2, B2, C1, D1. And the timing.
Yeah.
When did they start spreading out?
That's fascinating. These 4 groups split off from their Siberian ancestors during that deep ice age period. Then they expanded south pretty rapidly, maybe between 20 and 16,000 years ago.
And how did they move so fast? Weren't there massive ice sheets blocking the way?
There were inland. That interior ice-free corridor probably didn't open up until later, maybe around 14,000 years ago. So the speed suggests they likely took a coastal route down the Pacific coast.
Oh, that makes sense. Like island hopping, but down the coastline.
Sort of, yeah. It allowed for that quicker dispersal south.
So that initial settlement sets the stage. But then over the next, 10,000 years plus, things didn't stay static.
Absolutely not. Those groups became incredibly diverse locally. That original Native American ancestry itself developed a lot of internal structure. For instance, genetic models suggest that the ancestors of today's northern indigenous groups in Mexico, like the Raramuri or Huixol, split off from the ancestors of southern groups like the Triqui or Maya somewhere around 6,500 to 7,900 years ago.
That long ago. Wow.
Yeah, and then thousands more years of relative isolation, regional development, that created this really distinct indigenous genetic landscape across Mesoamerica before any Europeans arrived.
Okay, so you have this ancient, diverse indigenous foundation. Then comes the collision, the colonial crucible. When we look at modern Mexican genetics, it's built on 3 main pillars, right?
That's right. You've got that indigenous American base, then the European layer, mostly Spanish, of course, and an African contribution, too.
The African part is often overlooked, isn't it?
It is, historically. But the records show somewhere between maybe 200,000 and 500,000 enslaved Africans were brought to New Spain, mainly between the 16th and 18th centuries.
And does the genetic data reflect that today?
It does, but it's complex. Overall, if you look at the total genetic mix, the autosomal DNA, the African ancestry, averages out as fairly small in the broader mestizo population, maybe in the 2% to 6% range typically.
But it's not spread evenly. That's crucial, right?
Exactly. That average hides a lot of variation. Researchers found pockets, areas with noticeably higher African ancestry, especially in states like Guerrero and Veracruz.
Which makes historical sense. Those were major ports, major entry points during the colonial slave trade.
Precisely. The genetics lines up perfectly with the known history and geography of where African populations were concentrated.
Okay, now here's where I think this story gets incredibly powerful, almost personal. The difference between male and female lines of ancestry. this sex-biased history. It tells a very specific story.
It really does. This is probably one of the most striking findings. If we go back to that mitochondrial DNA, the maternal line, it's overwhelmingly indigenous American.
Overwhelmingly. Like, what percentage are we talking?
Studies consistently show between 85% and 90% of mtDNA lineages in Mexican mestizos trace back to those 4 founding Native American haplogroups we mentioned, A2, B2, C1, D1.
Wait, let that sink in.
Yeah.
Almost 9 out of 10 maternal lines connect directly back to the women who were there before Columbus. Through centuries of upheaval. That's resilience.
It's a profound finding, a deep continuity of indigenous motherhood. But, and this is huge, but the picture flips completely when you look at the paternal side.
Okay, so what happens with the Y chromosome, passed only from father to son?
The contrast is stark. Paternal lines are predominantly European, specifically Southern European, matching the Spanish colonial history.
How predominant?
It varies, but the European Y chromosome contribution can range from around 48% up to nearly 89% in different admix groups across Mexico. And if you look at the total genome mix, the autosomal DNA, European ancestry tends to be lowest in the South, maybe around 30% on average, and highest in the North, closer to 60%.
So if you put those two together, nearly all indigenous mothers, but a huge proportion of European fathers, what does that pattern scream about the colonial encounter?
It points directly to what geneticists call directional mating, or sex-biased admixture. It's a pattern seen across much of Latin America, actually. It reflects the social reality of the time. The primary mixing happened between immigrant men, Spanish, other Europeans, and local indigenous women.
That pattern isn't just a dry statistic. It's a genetic echo of power dynamics, social structures, conquest, and survival. It means the indigenous genetic identity largely survived through the female line.
Exactly. And that fundamental asymmetry shapes everything that follows genetically.
Right. And speaking of what follows, a huge takeaway from all this genomic work seems to be, you absolutely cannot treat Mexico as one single genetic blob. The diversity within the country is immense.
That's a critical point. The studies emphasize considerable heterogeneity. And it's not random variation. There's a clear pattern. A gradient, or cline, running from the north down to the southeast.
Okay, walk us through that. How does the north differ from the center and the south?
Well, if you start up north, states like Sonora, Zacatecas, the samples there tend to show the highest genetic diversity overall. We measure this partly with something called heterozygosity, basically, how many different gene versions you carry.
More mixing, more diversity?
Generally, yes. And genetically, these northern populations cluster a bit closer to European reference groups. This probably reflects historical settlement patterns, maybe later European migrations, and different indigenous population densities in those areas.
Okay. And then moving south into the historical heartlands.
As you move towards central and southern states, think Guerrero, Guanajuato, Oaxaca, the genetic profile shifts. People there are genetically closer to present-day Amerindian groups, like the Zapotec people used as a reference.
Which makes sense, given that's where the major pre-Hispanic civilizations were centered, and where indigenous populations are still densest today.
Exactly. The correlation is very strong, and the difference can be quite significant.
I remember reading something striking about that, a measure called FST, which gauges genetic distance.
Yes. They found the FST distance between, say, people in Sonora in the north and the Zapotec indigenous group in the south was actually greater than the distance between some distinct East Asian populations like Han Chinese and Japanese.
Wow. So the genetic difference within Mexico can be larger than between entirely different countries in Asia.
In some comparisons, yes. It just hammers home how much internal genetic structure there is. You can't just apply findings from one region to the whole country, especially for health research.
And what about the far southeast, the Maya region?
Also distinct. Mestizo populations down there, in the Yucatan especially, show a clearly identifiable Maya genetic contribution. The Maya had a somewhat distinct history and less interaction with Central Mexican groups for long periods, and that's visible in the DNA.
So this incredible journey, Ice Age migrations, colonial collision, leading to this complex, geographically patterned mosaic today. Let's shift gears slightly. Why does understanding all this history matter for someone listening right now? How does it connect to, say, health?
Oh, it's absolutely crucial, especially for what we call genomic medicine or precision medicine. We know that genetic variations affect everything from your risk for certain diseases, diabetes, heart conditions, cancers, to how you metabolize drugs.
And most genetic research has been
It's
done.
overwhelmingly on people of European descent. Something like 80% of the data in big genomic databases comes from European ancestry populations. That data just doesn't fully capture the variation present in admixed populations like Mexico's.
So if you use the wrong reference map, your medical predictions or drug dosages could be off.
Precisely. That's why projects like iNimagen and the Mexican Biobank are so vital. They're building a Mexico-specific genomic map. They've already shown that for predicting risks for some complex traits, using their local data works significantly better than relying on, say, the UK Biobank data.
So knowing where someone's ancestry likely falls on that north-south gradient, for example, could actually be medically relevant.
Potentially, yes. It helps refine risk assessments. It informs clinical practice and allows public health folks to target interventions more effectively towards populations with specific genetic predispositions.
Fascinating. And beyond disease risk, did these studies find any clues about how different groups adapted to their environments over time? Any signatures of evolution?
Yes, some really interesting ones. Unsurprisingly, some of the strongest signals are in immune system genes. Genes like IL-17A and IL-13 show signs of having been strongly selected for.
Which probably relates back to disease.
Almost certainly. Think about the massive epidemics introduced during the colonial period: smallpox, measles, huge population die-offs. The survivors likely carried gene variants that offered some resistance, and those variants got passed down, leaving a strong genetic signature. It's a stark reminder of that demographic catastrophe.
Wow. History written in the immune system.
Exactly. And you see local adaptations, too. For example, among the Triquer people in Oaxaca, researchers found selection signals in a gene called KBTBD8, which is linked to shorter stature. It shows how specific regional pressures can shape physical traits over generations.
So putting it all together, we have this incredibly deep history, starting with those first coastal migrations millennia ago, then this intense period of colonial mixing, marked by that stark difference between maternal indigenous roots and paternal European lines. And the result today is this rich but very heterogeneous genetic landscape across Mexico, varying significantly north to south.
And I think if there's one core message to take away, it's the resilience embedded in that maternal lineage. Despite everything, conquest, disease, massive demographic shifts, that indigenous mitochondrial DNA persists at nearly 90%. It underscores that the foundational genetic heritage of Mexico carried through the women remains incredibly strong.
It's an amazing continuity. And that final point about heterogeneity, knowing that European ancestry might be around 30% in the south, but 60% in the north, that's not just history, is it? It's a blueprint for the future in some ways. It raises a crucial question for you to think about. Given these clear regional genetic differences, how should Mexico tailor its public health strategies, its resource allocation, to actually address the diverse health risks revealed across its different geographic zones? That's the challenge these genomic insights pose right now.